In this video, I interviewed Jack Scannell. Jack is perhaps best known for coining the term Eroom's law. You've probably heard this if you've been in the biopharma industry for a while, but if you haven't, it essentially states that the cost of developing a new drug has increased exponentially over time, despite exponential improvements in efficiency and throughput at individual steps in the drug discovery and development process, like the cost of genomic sequencing has dropped exponentially over time, improvements in combinatorial chemistry and other areas have been dramatic, but the cost of developing a drug has just skyrocketed. Earlier this year, he published a paper that showed some evidence that that trend actually may be reversing, and we'll talk about some of the implications of that for startups. So a little bit more about Jack before we jump in. He was originally studied medicine, but then he told me he dropped out, and then he went to study academic neuroscience at Oxford. Then he worked in drug discovery at a small biotech. He worked in consulting at the Boston Consulting Group, and then he went to work in biotech investment at UBS and Sanford Bernstein. And he's doing mostly consulting and some academic work today, but he's truly one of the thought leaders on R&D productivity in biopharma and has a lot of very, very important insights in that space. So without further ado, let's jump into the interview.
To start out, it would be great to just have you define for us Eroom's law.
Well, sort of Eroom's law, the term was kind of a joke. I had probably for a decade, when I sort of first coined the term, been interested in what struck me as a very ugly contrast. So, I was a biological scientist originally. I, studied medicine, did a PhD during my medical training, never went back to medicine, but I sort of, I started my professional career thinking I would spend my entire life as an academic biomedical scientist. And so I've always been very interested in how cool science is and all of the brilliant, new, exciting things that are happening. But I ended up working in consulting and then working for an investment bank, being what's called a sell-side equity analyst, where I was looking at the economics of the drug industry. And I was struck by a horrible contrast in that all of the things that I, as a biological scientist, had been aware were getting hundreds, thousands, millions, or in some cases billions of times cheaper and better, didn't seem to stop the fact that in rough terms, the industry was spending 100 times more per drug approved in 2010 than it was in 1950. Right, so that struck me as a sort of huge rhetorical mismatch, right? Because if you looked at the individual technologies that were meant to be important for drug discovery, they were getting hundreds, thousands, millions, or billions of times cheaper, but the output was getting 100 times more expensive. And at the time, lots of people like me working in investment thought the average big drug company was losing money in its R&D, right? And that was sort of struck me as a sort of a contrast that demanded an explanation, as well as being one that was important for sort of policy, as well as kind of purely financial and medical perspectives, right? So that was the thing that got me interested. And Eroom's law, it was just a joke, right? It was in the inputs seemed to be obeying Moore's law, whether it was DNA sequencing costs or or computational chemistry costs, and the output was going in the opposite direction.
Yeah, I think that's really striking, especially when you consider how much cheaper things like sequencing have gotten. And that's an incredibly striking sort of figures when you look at it, just the cost of sequencing going down so much over time, and then the cost of chemistry as well, and just creating libraries. But then overall, this period where those phenomena were occurring was actually one of the least productive in terms of R&D efficiency in the history of pharma. So why is that? Why did all these great throughput increases that we were able to accomplish with new technology, why didn't that translate into better drug development R&D?
So I will qualify my comments before I answer that, and you might want to come back to this. Actually, things have improved a bit since 2010, right? So Eroom's law didn't hold, right, which is a good thing for everyone. But the improvement since 2010 is still kind of pitiful compared to the decline from 1950, right? So, yep. so let's not congratulate ourselves too much. I think the paper I wrote in 2012 was actually an attempt just to sort of provoke people into better diagnosis. So in it, we suggested a number of causes. And we also made a point, which I think is an important one, which is if you've got a process like drug discovery that gets 100 times more expensive when the inputs have apparently got hundreds or thousands or billions of times cheaper, there are two broad classes of explanation for that contrast, right? One is got some sort of catastrophic resource depletion, right? You've run out of something, right? So it's a bit like kind of biological barrel scraping. If the barrel's nearly empty, you scrape it faster and faster, you're still not going to get much out of it. So that's one broad set of explanations. And the other broad set of explanations is actually what we've done is we've optimized the wrong set of activities. So what we're doing is we're doing things at incredibly low unit cost and high volume, but those are fundamentally the wrong things to be doing, right? So the idea would be there's some kind of qualitative change that's happened in R&D that means that we're doing the wrong things. And those are the two broad classes of explanation. And the problem for people working in drug discovery is that the more you disagree with one of those, the more you're forced to agree with the other one, right? So it's a kind of unpalatable situation. Now, in the 2012 paper, we suggested a number of things, but I think we alluded to the idea that there might be some sort of misindustrialization, like people doing the wrong things. But I think the 2012 paper looked a bit more at what you might call those resource depletion ideas. And it highlighted one problem that I think is, it's not unique to the drug industry, but it's pretty unusual. And you see it in the drug industry and perhaps the crop protection chemical industry. And I don't know if you see it in many other places. And that's what we call the better than the Beatles problem. So the drug industry is unusual in that it's an intellectual property business where people don't get bored of the intellectual property. And the old intellectual property becomes this kind of huge economic competitor against the new intellectual property. So we called it the Beatles problem because there's like an analogy with Beatles records. And again, here I'm showing my age and lack of fashionability, right? But imagine that every new record had to be better than the Beatles, right? And you could download all the old Beatles records for free, and people didn't get bored of listening to Beatles, right? If you had those set of criteria apply, it'd be quite hard to make money out of launching new music, right? Now, drugs are like that. So metformin is a bit like the Beatles equivalent in type 2 diabetes. It's now generic, it costs cents a pill, it's incredibly cheap, it's incredibly effective, and there's also, by the way, 10-ish other classes of new diabetes drugs, right? So this means that any new diabetes drug, most of those classes are generic, so they're almost free. So any new diabetes drug has to compete against that stuff. And what that's done is it's pushed, it's very clear, if you look at what the drug industry is spending its money on now, it's pushed R&D into what's euphemistically called unmet medical need, but might be less euphemistically called diseases that over the last 100 years the pharmaceutical industry has been unable to solve, right? And for one reason or another, they're likely to be hard, right? However you define hard, if you haven't cracked it since, 1910, when people started doing modern medicinal chemistry, maybe there's something difficult about it, right? So that's one problem. Another obvious drag is regulation. And I think if I was writing the paper again, I wouldn't call it the cautious regulator problem, because actually I think the regulator is pretty good. But it's undoubtedly the case that in crowded therapy areas, regulatory barriers go up. So the things that would have got you approved as a diabetes drug or a hypertension drug 15 or 20 years ago or 30 years ago would not get you approved now, right? So those were two of the kind of resource depletion ideas. But it led subsequently to some more thinking about the technology idea. And I think that's probably what we're going to talk about mostly now. And you may want to direct me with a question, but that led to ideas about sort of predictive validity and screening disease models.
Right, and I think I'll just quickly kind of recap what you said, because I think it's incredibly valuable and very insightful. And I think maybe we can talk a little bit about some of the implications of that before we jump into the predictive validity discussion. But I think the things you mentioned in terms of the issues you called out in that 2012 paper, so the cautious regulator problem, which maybe it's a good regulator, and it's regulating things that are difficult to regulate in important human health. So it's warranted, but it's hard to get through that process regardless. And then the better than the Beatles problem, you have all of these great drugs that have been developed in the past, but now genericized. So something like 80
and 90% of US prescriptions are now for generic medicines, right? So it's just this huge, effectively free competition across most of the common diseases that affect most people most of the time.
Right, and a lot of those genericizations were occurring about the time where you wrote that paper, that sort of 2010 to 2012 quote unquote patent cliff, really had a flood of genericization. So I would imagine that, coupled with Eroom's law problem overall, posed great challenges to the industry. And then you also mentioned the the tendency to either throw money at things to try to fix them, or another thing I think you called out in the paper was the basic research sort of brute force bias, which we'll talk about a little bit more. But I think first we'd love to touch on what you mentioned in terms of how the situation has changed. And you put a paper out earlier this year about breaking Eroom's law. So can you just walk us through a little bit what has happened since 2012 and why that's important?
Yeah, so we were cautious actually in the 2012 paper, and we said Eroom's law may be broken because there's a few things happening, right? So we could see down the pipe a little bit, and other people could as well. It wasn't just us. I think the molecular slicing and dicing does a number. So again, sometimes branded as personalized or precision medicine, right? If you get better at subsegmenting patients into specific groups with specific diseases, it means your disease models work better, because if you can distinguish ALK positive non-small cell lung cancer from other non-small cell lung cancers, actually you could make a reasonable screening and disease models of ALK positive non-small cell lung cancer. So you have these tools to match effectively what's coming out of the pipe with the right patient population, right? So you have more diseases, and you can better broke the match between compounds and diseases. I think that's helped. I also, arguably, I think part of the driver here is economic and part regulatory, but the industry in the sort of early 2000s and since 2010 has discovered quite how much it can charge for orphan drugs, right? And I think lots of very rare diseases tend to be monogenetic, and monogenetic diseases are more technically tractable, both more modelable, and the regulatory hurdles tend to be a bit lower. And then also, I think there's been a lot of what you might call sort of innovative trial design in sort of phase one-ish oncology trials, again, which mean you sort of add almost kind of experimental medicine in man and if you combine that with the right diagnostic technologies, you get a much better opportunity to find molecules that do something for some people, even if you did a big trial in an unselected patient population, you wouldn't see it. So I think those things have sort of worked together to mean that we've had more coming out the pipe.
Yeah, so this combination of essentially increasing the predictive validity of the model by defining it to a more narrow and easier to model subgroup. And then also, again, targeting these areas you said of unmet need, where if it's a rare disease that hasn't been, the pharma industry hasn't paid attention to it, you're not going to have that better than the Beatles problem. But I think maybe before we go too much further, I feel like we're saying predictive validity a lot. So maybe we should define that really quickly for everyone before we jump into the next topic.
So after the 2012 paper, I did a few things for a while, but I was still very interested in the idea if there was sort of technology related issues in terms of the drag on R&D productivity. And for reasons I won't bore you with unless you desperately want me to, I sort of started trying to apply sort of formal search type models, right? So I said, well, if we treat R&D as a formal search process, what parameters would this search process be particularly sensitive to? And you can, I think, in a fairly sort of common sense way, think of R&D as a sort of search problem, where there is either a real or often a sort of notional universe of therapeutic candidates, and you could think of that as the drug targets we might choose to pursue, or you could think of it as the compounds we might conceivably synthesize against a particular drug target, right? And then what you have is you've got a bunch of tools which help you decide whether those targets or whether the compounds you've synthesized against them are likely to do anything useful in a human, right? And if you then try and formalize that, you have to have a formal measure of the degree to which the tool can distinguish between good, useful, and more useful and less useful therapeutic candidates. And that is what I call predictive validity. And in most of the formal analysis I've done, I operationalize it as a sort of notional correlation coefficient. So you say, well, okay, if I had infinite money, no ethics, and no regulation, and I could get all of the therapeutic candidates that I might conceivably ever make for this particular disease, and I test them all in a particular disease model and get the score from the disease model, and then I put them all into a phase 3 pivotal trial. And then I plot a graph where I have the endpoint score in the phase 3 trial against the score in the rat or the high throughput screen or whatever it was. Predictive validity, I operationalize as the correlation coefficient that exists, but it's a formal measure of the degree to which the output of your model matches therapeutic utility in man. And one could operationalize it in a number of different ways, but I typically use correlation coefficient.
Got it. So it's basically how well do your disease models predict how well the drug is going to work in humans. Yeah. And right now, the way that people test that is by spending tens or hundreds of millions of dollars on development and clinical trials to see if it works. And then I think the 2010 Paul et al. Paper showed that I think 30 or 40% of drugs failed in phase 2 for reasons largely of effectiveness, obviously. And I think that was an interesting paper because it showed that the biggest driver of the cost of drug development was the risk of failure in phase 2 or 3. Which definitely corroborates the finding that you had and illustrates the importance of good predictive models. So how important are these as this predictive validity in terms of the sort of fundability of a drug development program?
Okay, so I'll be honest with you first, right? Predictive validity, I would say it's a bit like the dark matter of drug discovery, right? So it's important. It exists in principle, but actually it's almost impossible to see or measure, right? Because actually we don't have infinite money, no ethics, and no regulator. Right? So, and there's another important point which may sound a bit geeky, but is actually the only things we put into man are the things that our model says will work, right? So there's a huge informational problem, which is lots of the things our model said won't work might have worked. Right. So actually you can't estimate true positive and false positive rates out of what we know about drug discovery, because people never see whether the negatives from your screening model are indeed negatives, right? So anyway, so it's this construct. It says, okay, we can imagine that there's this correlation coefficient that exists between the output of our model and the human outcome of interest. There is very good suggestive evidence that suggests in cases where R&D has been a total and utter failure, the predictive validity has been low, and in cases where R&D has been very successful, it's been very high, but it's actually impossible to prove precisely what it is, right? And I'll give you a couple of specific examples. Or actually, probably the nicest historic example I know is antibiotic R&D. So the world's first useful antimicrobial drug the first useful antimicrobial drug of the 20th century was a drug called protonsil, or sulfanilamide. And this came from one of the German chemistry/drug companies circa 1930. And a screen of around 200, or a screen and then optimization involved around 200 compounds, led to the discovery of sulfanilamide. So the throughput was very low, n equals roughly 200, right? And we got a useful antimicrobial, which until penicillin came along was probably the most useful, broadly useful antimicrobial there was. Now, between 1995 and 2005, the global drug industry decided to use high throughput screening to try and find broad spectrum antibiotics using targets that were identified doing clever bacterial genomics, looking for essential genes. And they mounted around 100 high throughput screening campaigns, some of which tested 500,000 compounds, and between them tested well over 10 to the 7 compounds. And they found not one single drug that was worth putting into clinical trials. Oh. Now, so the question then is, well, how is it possible that someone can take 200 dye stuffs and dye stuff derivatives, which is essentially what he was using in 1930, test 200 of them and find something useful, but in 1995 to 2000, we can test 10 to the 7 compounds and not find anything worth putting into clinical trials? Well, there is a bit of better than the Beatles problems at play, right? The regulator got tougher as well, but it turns out that, or rather, in 1930, Gerhard Domach, who was doing this, was screening his compounds in mice with sepsis. So this was a whole animal in vivo sepsis screen. we now know that high throughput screening for potential antimicrobials does a whole bunch of things to decorrelate the output of your model from the human outcome of interest. So, for example, high throughput screening collections are enriched for compounds that don't get into bacteria, right?
Mm-hmm.
The genes that are essential for bacterial survival in culture are not necessarily the same as the genes that are essential for bacterial survival in vitro, right? So you can actually start mapping out the factors that mean, despite the fact your throughput went up by a factor, by 10 to the 5, you've actually reduced the validity and made the R&D process less effective.
Right. So you basically have these models that are built to be high throughput, but they lose so much information in the process of scaling them.
And if you run the decision theoretic maths, I mean, it varies a bit on things like how rare positives are, right? But if you assume positives are rare, and by rare I mean in your universe of drugs, a randomly selected thing would work in a human less than 10% of the time, right? Under those circumstances, very often you'll find that an absolute 0.1 change in the correlation coefficient between putative model output and the human outcome of interest has a greater effect on your productivity than, for example, doing 10 times more experiments, or sometimes doing 100 times more experiments, right? So the predictive validity parameter, this correlation parameter between the model and the outcome of interest in man, is actually a very, very powerful parameter compared to, for example, your ability to put more and more items through your R&D process.
Right, and I think that really kind of leads to an interesting question, is that you have these small differences in the predictive validity that are so important to your overall R&D productivity. But as you mentioned earlier, predictive validity is sort of this nebulous concept that in practice is almost impossible to measure, especially for new models where you don't have a lot of clinical evidence. So how do you go about taking this potentially incredibly useful information where you have these very small changes in this value that are incredibly important to your program, but are really hard to measure? Can you even measure that? How do you go about trying to tease that out?
So I'll tell you what I'm trying to do in practice, right? And who knows whether this will have any success at all, right? But this is what I'm trying to do. I think the first thing you need to do is you actually need to educate your audience on the decision theoretic maths, right? And as I say, the maths is not particularly exotic. It is a pretty straightforward implementation of the way most people think R&D works, right? But you have to convince people of the result. That small changes in predictive validity are very, very important. And if you convince them of that, I think the sort of narrative story is something like this. It says, look, most of the time, if it's a disease area for which we don't have lots of useful drugs that people haven't been working on for the last 70 years, you will never know the predictive validity of your models for sure, right? But you can be confident that small improvements in validity have the potential to have very, very large effects on productivity, right? Therefore, you need to do everything you can to sort of nudge this nebulous thing in the right direction. So how do you do that? Well, I think one of the first things you need to do is you need to think a bit hard about how you evaluate validity, right? Or proxies of validity. And there's a number of them, right? So one set of things, not very exciting, is what you might call experimental hygiene. So there's quite a big furore at the moment in the academic literature about irreproducibility, right? And there's actually now quite a nice large literature. A lot of the best of it comes from stroke, which was an area where R&D was very unsuccessful for a very long time. So they looked at it very hard, ischemic stroke. But there's now actually quite a large literature on the sorts of things you should do and shouldn't do if you want a sort of a good hygiene factor from your models. So, for example, are you having blinded assessment of animal model data, right? Are your sample sizes big enough? Are there other sources of bias? For example, there's lots of publication bias, right? So there's sort of literature around statistical hygiene, or rather experimental and statistical hygiene. I think lots of people in the drug industry know that, right? But I think in academia, it's not known as well as it should be. I think there's then what you might call an evaluation around biological plausibility, or how similar your model is to the disease state in man. Now, the academic literature on that point is surprisingly small, but there are one or two very nice frameworks that I think can be a starting point for effectively doing some kind of biological evaluation. So, and actually, we talked in a bit more detail about how practically one should do it. But essentially, you need to get suitable experts to say, for this particular disease, here are the things we would really look for in a model, right? And it's going to vary from therapy area to therapy area. It's going to vary from whether it's an in vitro model to an in vivo model, right? But you need some kind of idea about what a good model would look like. What are the things that we think would predict pharmacological predictiveness, right? And then you evaluate your model and other models against them. I think there is then something around what you might call, or actually there's a guy called Frank Sams Dodd who's written quite nicely about this. He talks about disease models. I've just talked about a disease model. Does the system you have replicate the biology that you think is important? He then distinguishes disease models from what he calls tests, right? So a disease model might be, for example, I've been looking at rodent models of stroke lately. This would be a model where you embolize the middle cerebral artery in a mouse, right, to simulate a stroke, right? That would be the disease model. But he then talks about tests. So the test would be the set of protocols you use to test drugs within that disease model and get an output. So, for example, it would be, we give the drug 15 minutes after we've induced the ischemic stroke, or 2 hours after we've induced the ischemic stroke. We then wait 5 days, we do an MRI scan, and then we wait another 3 days, 2 weeks, and see what sort of behavioral recovery you see, right? So it's the mechanism by which you try and simulate the human pathological process and the measures you apply in that disease model, right? And then I think the fourth thing you need is very clear thinking about what I call domains of validity, right? And this is something that is certainly remarkably absent very often from the academic literature. I think in industry it's thought, people tend to think about it harder. But the best illustration of domains of validity actually comes from physics. A former colleague of mine who's a physicist by training, I had no idea what domains of validity were. I was a biologist. We didn't talk about this. But about 5 years ago, I had a physicist explain to me. He said, look, a physicist will know that Newtonian mechanics is really good for predicting things that are quite a bit bigger than an atom, going quite a bit slower than the speed of light. So if it's bigger than an atom, slower than the speed of light, you can use Newtonian mechanics and you get the right answer. But if it's smaller than an atom or close to the speed of light, don't use Newtonian mechanics because you get it wrong, right? So it's the set of conditions under which you think your model predicts things, right? So I think if you have the hygiene factors, right? If you have some evaluation of biological plausibility, if you have a test that is the right test, i.e. you're actually getting your model, which may recapitulate human pathology, to actually do something that resembles the human disease and the way you measure the human disease, and if you understand the domains under which all of this is valid, I think those are the sorts of things you should have in order to decide the degree to which your model will be more or less useful.
Gotcha. So it almost sounds like just being a lot more kind of intellectually honest about what you know and what you don't know about a model. How good is this model recapitulating disease? How kind of finicky is it if you have different protocols?
Yes.
With the domains of validity, like
right.
what
I think exactly. I
can you
think
draw?
a starting point is very thoughtfully constructed checklists, and then honest evaluation against those checklists. That's a starting point, right?
Mm-hmm.
Because unless you can sort of articulate it and think about it in concrete terms, it's then quite hard to sort of manage it.
Yep. And it sounds like those kind of things would be very difficult to implement at scale. Like if you're a large pharma company and you want to go through all of your discovery programs and R&D programs and implement this sort of rigor, that sounds like an incredibly challenging organizational task. Is that the case, do you think, for a large
I think
company?
it would take a long time and be difficult. A lot of you'd have to be very motivated to do it, I think.
Right. And I think that kind of the second part of that point would be if you're a smaller company or a startup or like a venture creation firm, you have a smaller team of people, and maybe you're able to essentially operationalize those principles a little bit more easily, because if you have a handful of people and you all sort of get on the same page about what's a good model and what isn't and how you're going to evaluate these things, I mean, maybe that's a little bit easier to operationalize it than if you're a big company. Is that?
I think that may be right. Partly just because you don't have the sort of legacy way of, you don't have the way you've always been doing things and the way you've got thousands. You know, it's not like you've got thousands of scientists and portfolio management processes and sort of reporting structures, all of which tie you into a particular way of working.
Right, right. But on the other hand, it does seem like there's a paper you cited in the Breaking Eroom's Law paper from earlier this year from AstraZeneca. I think we talked about before, and it did talk about their ways of operationalizing these, and they showed some data on the organizational change in performance, and it had a lot more things that were killed early in the target, sort of for target validation reasons, and they had a lot more, which I thought was really interesting, a big increase in target identification efforts and a decrease in lead discovery and lead optimization efforts, which I thought was very interesting.
And I think there are a number of companies. It's always difficult, you know, looking outside of big drug companies, right, to know precisely what's going on. But I'm pretty convinced that you know, there was a major change at Astra, partly triggered by, you know, having been through a very, very difficult period in terms of R&D, right? So effectively, they were really motivated to do something different. And I can read those Astra papers, and you know, if I sort of have a little checklist and sort of use a sort of predictive validity lens to look at what they're doing, lots and lots of the things they say they're doing look to me very much like the sorts of things one should be doing if one was arguing from predictive validity lens. I think they've used different language, right? They communicate about it internally in a different way, but it's pushing things in a very similar direction, right? Effectively, most of the things they talk about are things that will effectively increase the correlation between the information they're looking at on which to make decisions and whether things will actually work in humans.
Right. I think they mentioned, cite a couple of your papers on that as well, but it was really striking to me, and it was also very interesting for me as someone who's not in the big pharma industry to see that sort of disclosure on their R&D processes. This is a very interesting data set.
And I think there's a sort of smaller company, although you know, Vertex are not a small company anymore, but I think there are also some companies who have what you might call a kind of validity-centric, or have had what you might call a validity-centric strategy, i.e., again, they've articulated it differently, but they've had a very clear idea about the kind of problems that are going to be tractable and the kind of problems that aren't going to be tractable. And they then go after the problems that are going to be tractable, right? So you say, well, look, unmet medical needs sounds great, but actually most of it is unmet, perhaps because it's intractable, right?
Yeah.
So can we within that find isolated little niches where actually it's going to be tractable, and that is sometimes based on arguments around, you know, clear genetic validation, you know, availability of human tissue, et cetera, et cetera.
Yep. Yeah, it's very striking that amongst the top 10 causes of death, at least in the U.S., you know, oncology or cancer is number 2, and that receives a lot of investment. But then a lot of the other ones are really, you know, haven't received that much investment. Obviously.
And oncology still has. I mean, it's very hard to get safety fresh data, but although there's lots of oncology approvals, it still seems to have very high clinical trial failure rates, right?
Yeah, it may even be exacerbated by the pure number of things that are going into the clinic these days.
Yeah.
But so I think before we jump into some of the technologies that, you know, AstraZeneca, but not just them, the ones they mentioned in the paper, but other startups are using. So you talked a little bit about this in the Breaking Eroom's Law paper. Do you think that this is a durable phenomenon? Do you think that we've sustainably reversed this trend of increasing, you know, R&D costs, or is it just a blip and we're going to go back to the same old Eroom's Law in a few years?
I actually suspect that we've got a period when I don't think approval is going to take a downturn. It's a very good question. It's actually not one I've thought about a huge amount. I think some of the things we've got now are kind of here to stay, right? I think sort of molecular slicing and dicing of disease, you increasing rare disease focus. I'm also, again, outside of my area of expertise, but I'm also impressed by what one could argue is actually there's quite a lot of new sort of therapeutic modalities coming down the line, right? And so again, if I think back to my sort of equity analyst days, which ended about a year ago, right? So I'm not super current. I think the thing that I was concerned about then was not that not many things are going to come out of the pipe. It's that competitive crowding actually seemed to be the problem, which is a better problem to have than you can't discover anything, right? Yeah, but it was an enormous amount of the pipeline is focused on bits of oncology and rare diseases, and some of those conditions are getting, that. So I think we've got a slightly different problem now, which is that the industry has learned that it can do well in areas with strong human genetic validation, right? And good sort of molecular diagnostics, right? But that tends to be pushing lots of people into the same areas.
Yep. Yeah, and oncology is obviously very competitive, and it can be difficult to recruit clinical trials, and getting commercial differentiation can be tough. And even for some of these rare diseases, I think that's some of the most exciting stuff that I see, just in terms of the dramatic clinical benefit that you can see in some of those disease areas. But there's some diseases where you have a few hundred or a few thousand people a year, and there's a couple dozen development programs that are going after those. And all of that is great for society, I think, obviously, because you have more competition, you have lower prices, more innovative medicines. And the question is for industry, like, is that sustainable? Are there other areas that we can explore? I think the point about the novel modalities too is very interesting, because it seems like it could be the very early days for those, where a lot of those novel modalities are going after a few oncology indications or rare disease indications, or certain tissue types. And the cost of making and distributing those is just so high. If over time we're able to decrease those costs, that could theoretically open up a much broader set of diseases to being treatable with these new modalities. So I think that's a very, very interesting area, and definitely one that I'm interested in when watching. So with respect to some of the technologies that we mentioned, and how those might impact the future of R&D productivity, it'd be interesting to talk about a couple of those and how they might improve the predictive validity of R&D programs. So I think that a couple examples are using human tissue, or iPSC derived tissue or cells to screen compounds. Another area would be just in general using genomic data or other sort of omics data to identify and validate targets computationally. And then I think phenotypic screening and target deconvolution is another interesting area, whether it's using or developing new phenotypic screening models, or new ways to figure out what targets are involved in hits that come out of phenotypic screens. But before we dive into this specifically, just in general, how do you think about a new technology and figuring out whether it is enough of a potential improvement in predictive validity to justify investment? Like how would you think about that?
So I'm going to say something quite depressing first, before maybe trying to be a bit more positive, right? So the depressing thing I'll say is, if you look at the history of the drug industry from 1950 to today, I still think the most amazing thing is how much better, apparently, the science and technology has got, and how it still costs about 100 times more to bring a drug to market as it did in 1950. So what that says is, there should be a fairly strong Bayesian prior, just from history, that most exciting prognostications about better drug R&D technology don't have, it's just that history shows, right, that most of the time, if someone stands up to you and says, this is great, it's going to make R&D much more productive, most of the time people are wrong. Right?
Yep.
And so, having said that, right, I think, what can I say that is more positive? I think I'm going to sort of say some almost anecdotal things about human tissue, right? I think that there are a lot of organizational, I mean, they feel pragmatic, but actually they're not, right? There's a lot of sort of organizational inertia around quantity over quality and what reasonable unit costs are for an R&D project. I think the decision theoretic work I've done seems to show that. Let's suppose someone comes to their boss in a drug company and says, you know, I've looked at the science here, I've looked at the models that are available, as sort of two ways we can do this. We could, you know, test thousands of compounds, or no, well, not thousands, you test maybe tens or hundreds in the rat model that we've been using for the last 15 years, but actually which we know all about it, but we know it's not very predictive, right? Or we could go through the really difficult slog of getting human tissue that we know is really hard, and it's difficult to get it in the right condition, and we have to scrat around, and we've got no idea how long it's going to take. And quite frankly, I can't tell you how many compounds we'll be able to test in it, or whether we can run a program. But my biological intuition and some back of the envelope evaluations I've done that I won't show you now suggest it's probably more predictive than the rat, right? The problem is that the correct decision probably would be to do the difficult thing at low volume and low throughput. But actually the decision that it's much easier to make is the one to do it the way you've done it already, right? So I think that's a real practical challenge. And you know, and I know, and I don't know enough about them in detail, but I know, for example, there are a number of large programs going on where people are trying to collect and bank and use human tissue. But my view is for a bunch of applications, it's absolutely probably the sensible thing to do, even if it's much more expensive and gives you much lower throughput, right? Organizationally difficult, but probably very important. Sorry, what was the next question?
Yeah, so one of them was kind of around like iPSC derived cells versus actually collecting tissue, and then also phenotypic screening. So
okay,
how does that?
So iPSCs again here I'm going to be a bit of an evaluation zealot, right? So it seems to me, both commercially and academically, there's a lot of evaluation by assertion, right? So maybe I'm being unfair here, but if I were a cynic, particularly reading the academic literature, the word valid applied to model is an almost meaningless term, right? That carries almost no information at all about that model. And again, to be uncharitable, one reads papers that one imagines are done as the following, right? So you find a new way to make a model, you make your model, you then notice that some of the features of your model resemble the human disease state a bit more than other models, right? And then because of that retrospective noticing, you post hoc say that your model's been evaluated, right? Imagine if we applied the same rigor to evaluating models that we apply to drugs in clinical trials. How would you evaluate a model? What you would do is, before you had shown them your model, you would get a group of independent experts and you would run a formal process with them where they would opine on the criteria that they would use to decide whether or not the model was valid, right? What you would then do is you would then get a separate group of independent experts to hold up your model against those criteria, right? And only at that point would you declare whether your model was more or less valid than anyone else's, right? And again, I think with lots of the new screening technologies, you have evaluation by assertion, right? So I don't know enough about the particular iPSC screening technologies to know the extent to which they have proven themselves to be better or proven themselves to be more like the human condition versus there is a reasonable hope, right, that they will be. But my view is actually we can do better than reasonable hope. But at the moment, I think often the financial incentives are not right for people to do better than reasonable hope. Reasonable hope is more commercializable sometimes than expensive evaluation that may actually say that your technology doesn't actually look much better than something else.
Right. And yeah, you're saying that there's things you can do with these iPSC-derived models where you work with them and certain things look good, but other things may not look good. They may not fully recapitulate, the cell biology that you want.
No, I'm absolutely prepared to believe that lots of them will look much better than the alternatives for a whole bunch of reasons, and will be better. But what I'm saying is we've had, the standard way of evaluating or declaring a model to be valid is the person who owns the model sets the criteria, right?
Right.
And then they declare that they have won on those criteria.
Right. And
that's before 10 years before we've actually know whether the predictions have proven right in the clinic.
Right. And then to your earlier point about kind of the 4 things you would do to better assess the validity of a model is just having independent experts figure out what would you need to see for this to be good, what experimental design would you want to see, what outcomes would you want to see to validate this model.
How reliable are you? Is it reproducible? How big are the error bars? All the things that, and then how biologically similar is it? Can you replicate the endpoints that you might replicate in a to what extent? And then do you understand precisely the domains of validity, the conditions under which you think it is predictive, and then those conditions under which you think it isn't.
Right. So it's almost a sort of, rather than saying we're going to opine on how good this is as a predictor, we're going to try to filter out the ones that we know are probably going to be bad if you have a more rigorous lens on them. Yeah. And I think that's probably what a lot of the funds do. If you're a venture creation fund and you have the technology you're evaluating, one of the things you would want to do is kind of put it through that ringer before you actually, move forward and fund that. So that sounds like it's sort of a useful and practiced approach, but one that maybe isn't as standardized or generalized as would be ideal for really moving this along.
Yeah, and I don't want to suggest that people don't think really hard about models, right? Because in a sense, they do, right? It's obvious that everyone working in the drug industry knows that arguably, model validity is the single biggest driver of R&D productivity, right?
Yep.
So in a sense, people know it's important. I think the argument I make is they don't. So I think what I'm arguing is that the thing that everyone already knows is important, which is model validity, is even more important than everyone already knows, right? So it would be preposterous to suggest that people don't think really hard about this. But I think it's so important if you run the decision theoretic maths that people need to be thinking a bit harder, or some people need to be thinking a bit harder than they do now.
Yeah, in the sense that this is the thing to focus on. And if you don't have this figured out, it doesn't really matter as much necessarily how much you can scale something up or how quickly you can move something forward if you don't have this piece figured out. Yeah.
If positives are rare, bad models are false positive generators. That's effectively what they are.
Yes, yeah, that makes sense. Yeah. And I think another thing I really like about kind of what you're working on now is a lot of the people I talk to, they really, as you said, recognize the importance of this problem and have their own ways of figuring out how to improve predictive validity. And a lot of it is around just, genetically defined or more homogeneous patient populations, or sort of doing more rigorous evaluations of models, or really trying to figure out what kind of biomarkers you can use to sort of better predict target engagement and things like that as you move through different species. But I think that you're working and trying to develop like a broader framework for this, and the more sort of elevate the importance of more formalized criteria for evaluating this. It just seems like it could do a lot more to move this forward, rather than people just kind of in their own camps doing their own things. It works really well for them, but if you have a whole field that's kind of moving along in the right direction, I think that would be great. And I think there's a lot of, at least out here in the Bay Area, a lot of startups really love the scale approach. Right? There's all this you can do with scale. You can generate massive data sets, analyze them at a scale never before possible. Have robotic platforms that can analyze many more experiments. Yeah, sorry, go ahead.
And I said I was going to be depressing, and I might eventually say something a bit more positive. And I think you've given me a cue to say something a bit more positive. I think I am very sympathetic to the idea that if you have large, sort of ways of measuring model systems and the human disease state on a number of dimensions, and if some of those measures are objective, right? Now there's not going to be any one thing, right? But for example, if you combine sort of some comprehensive mapping of gene expression profiling with some good way of looking at sort of whether the histology looks the same, and whether various other parameters resemble a human state, the more of those things you have and the more they line up, I think the more confident you can be, the more confident, or rather, the more rigor you can put around the argument that somehow we're recapitulating the human disease state. And we're not just simply selecting one or two features which superficially look the same in our thing and man, but actually which are not going to predict any, which are not going to predict pharmacology. Right.
Right.
And also I'd say something else, which is it's not a result from decision theoretic work, which I emphasize as much, partly because I think it's a secondary result. I think it's an important one, which is if you have a mixture of sort of informatic and wet approaches to look at things, right? You don't just have one model. And if you've got a number of different models, it's important to understand the degree to which their outputs correlate with one another. And I'll give you a concrete example. People who do early drug discovery do this thing called counter screen. Probably the best way to explain it is this. Most biologists, right, if you run two different models which are meant to predict utility of the same set of drugs, and those two models seem to give you completely non-congruent answers, you would regard that as a problem, right? And in fact, there's quite an interesting spat in the academic literature about the degree to which different cancer cell lines from the NCI give you the same results when you put different drugs into them, right? So the non-congruence between slight different implementations of screening potential cancer drugs in cancer cell lines became a bit of an academic controversy. But actually, I think the informatics folks probably know that non-correlated models can actually be much more useful than correlated models, right? And if you've got two models that give you the same answer, formally they're redundant, right? Actually, the second one doesn't add the information. But if you have reason to believe both are valid and they give you different answers, that's actually much more informative, right? So these, and I think some of the sort of AI type firms, or the firms that have these sort of big data approaches, I think they probably have more, or in principle, one might see more sophistication in the way that information across different models and different platforms is formally combined, right? And the degree to which you measure whether or not the different models you've got are orthogonal and therefore potentially complementary with one another, or whether they're merely redundant and actually they don't add any information.
Right. And I think that's the fact that you can do so much more with scale. I was kind of saying earlier that maybe that can be an issue if you don't also have that other piece that you mentioned of validating your models and then also validating the congruence between the different models you use. And I would imagine, and I know some companies are doing this, if you have all of the best sort of new technologies for doing this high throughput screening and high content imaging and omics, which is incredibly expensive to get up and running, but if you can put that together and you think really hard about what you're actually doing in the wet lab and how good or how not good your models are and how much it can tell you or can't tell you, then that could be, I would think, a very, very powerful tool to generating new targets or new hits and new insights. But so if you're a company like that has sort of in its DNA, computation, scalable biology and informatics, how would you advise them to think about validating their models and making sure that they're able to look at the results of these high throughput screens with the appropriate level of confidence in what comes out?
I'm going to say one very general thing and then one very practical thing. The general thing is I think if you've got companies sort of computational, particularly sort of AI, sort of machine learning type backgrounds and expertise, there'll be certain methods and formalisms around model testing and model evaluation that some people in those companies will probably have, right? But I still think that the way you try and evaluate predictive validity is going to be broadly the same, right? You think about experimental hygiene, right? So are the error bars big? Is it reproducible, et cetera, et cetera. You think about biological plausibility. you think about what I would call the test, right? Whether endpoints and protocols match the human outcome of interest. That may be difficult in early drug discovery, right? But it gets more and more relevant as you get later. And you think about domains of validity. So that's a sort of general thing. I think generally it's similar. The one very specific thing I would say, and this may be too narrow and anecdotal, but I have had experiences in the past where informaticians have been insufficiently sensitive to the nature of the way the data that they're crunching is being generated, right? So, and this is going to vary particularly by the informatics domain, right? But in some informatics domains, the underlying data quality can be very, very poor. So what you might find is you might find people applying brilliant computational methods to data that is actually garbage, right? And yeah, I'm sure that's not true at a lot of places, but I think it is also true at some places. So I think you really need to know where the numbers you crunch come from and how they're generated.
Yep.
In gritty, biological, messy, technical terms.
Yep. Yeah, I think a lot of companies are moving towards that sort of in-house biological data generation and analysis, which is great in terms of validity, but obviously in terms of cost and setups that can get really expensive. I think one really interesting area to think about is you have these large-scale platforms and these techniques for generating valid data in-house, and then all these other technologies like omics and high-content imaging that can be super expensive if you add them up, and they can give you amazing data that you could never get before. And if you can evaluate that data properly, it's really exciting, but it's super expensive to get it up and running. So I don't have really an answer for this, but the question of like, what is the right amount of spend at an early stage to, what's the balance between spending on a really high throughput platform and getting all of these different assays up and running, and not wanting to just over invest in that platform to start. And I don't really have an answer to that question. It's sort of just putting it out there. I think it's an interesting area. How much is too much to spend on these things versus what you get.
Yeah, I don't have any strong views. I think one thing I would say though is that I think appropriating the value of models is difficult, right? And that's probably a separate conversation. But I think part of the challenge that the industry faces, and part of the reason we're in the situation we're in, is that you make money in drug R&D from patentable compounds. And very often, if you invest to create a better model that leads to patentable compounds, the minute those compounds show efficacy in phase 1 or phase 2, more likely, effectively, other people could make similar compounds and can get value without having gone through the investment in producing the model. Right? So I think that's a broader problem. But I don't have a particular answer to your question.
Yeah. I know it's different funds that I talk to have different approaches with respect to how much you'll invest in things like this, and I think it's an interesting open question. But yeah, your point, and I know you think a lot about this too, is your point about you can't actually get protection on these models and these platforms. You can predict what comes out of it.
Well, even if you could protect the model, the minute you got positive phase 2 data from your drug hitting target X,
right?
Everyone knows they can hit target X, even if they hadn't made the investment in the model that let you be the first person to know that you could target X. That's
right.
The issue.
Right, and yeah, I've definitely seen cases before where if you're better at developing a better compound, even if you don't develop the target at all, you can just piggyback on other people's efforts and get something with better distribution or potency, and then you win. So that's also an interesting dilemma. Well, and I know that you think a lot about that. It's a topic for a different discussion, but I don't want to take up too much more of your time. But I thought this was a super interesting conversation. I think it'll be very relevant to a lot of the people listening.
Okay.
And yeah, I've always just personally been an admirer of your work. I'm coming from the more sort of finance and economics side of biopharma. So this whole like evaluation of the efficiency and productivity at the higher level of R&D has always been an intellectual interest of mine.
Right.
And yeah, definitely have read your papers many times. So very grateful to have you on the podcast and have the chance to speak with you about these things.
No, it's very kind. I hope you and whoever listens to this finds it interesting. Thank you very much.
And before we wrap up, is there anything that, if people want to follow your work, is there anywhere where they can do that? And then I also want to ask if you have any recommendations, sort of recommended reading on either literature or books on the topics we discussed.
So maybe not altogether. I mean, it won't be surprising actually. I'm trying to write about this at the moment. So can people follow what I'm doing? Well, I don't have a particular social media presence, and I'm doing quite a lot of work on the things we talked about, and I hope to publish it in some fairly chunky papers. So, and it'll be difficult to talk much about those until they come out, or indeed if they come out. It does so happen, though, that in writing those chunky papers, I have actually tried to put together a sort of what I would call a sort of predictive validity reading list. And what I'll do is I'll send it to you, and then you can sort of maybe post some of the highlights along with this interview so that people can see it. I'm very happy to do that.
Oh, that would be great.
I have been giving a lot of thought, and now I'm getting very old. I think I might even have called it a young person's guide to predictive validity. Right? So it's essentially the things that I wish I'd found and read years ago, but now I know they exist.
Oh yeah, that's awesome. I've been looking for a reading list like that. I know others have as well. So I'm glad that I asked that question. Awesome. Okay, well, great. Well, thank you again for taking the time to do this. And I'll post all the links to your papers and the ones that you send in the comments to this. But this was a lot of fun. Thank you. Thank you so much.
No, thank you very much. Enjoyed it.